Hi, everyone. Welcome to my backyard behind me. Trying to get away from my kids who are, I think, trying to watch every single video that's ever been put on YouTube right now. So I'm outside, and because of that, I'm wearing my Beats headphones to hopefully get a bit better voice recording for you guys. Plus, makes me look like a DJ. So, bonus. Um, so even though I'm outside, cup of tea, it's always really important. And what we're going to talk about today is chapter 8, variation in chromosome structure and number. Now, I want you to make sure that you read the reading guide or study guide that's on Moodle for this, this chapter, because, for example, we're not going to delve into 8.7 at all in this chapter. So the first lecture is just going to cover the first 3 portions of the chapter. Then we're going to do another one for the last few portions. So to date in this class, we've mostly been talking about what I would call allelic variations when we've talked about differences in DNA sequence from one organism to the next. So one gene that has different alleles in various forms. Today's lecture, or today's section of the class, I guess, is dealing with large genetic changes, which means often hundreds, even thousands of genes. In fact, we're talking about changes that are so big, you can actually see them with a light microscope in order to see them. So very large changes. These are so-called chromosome aberrations. And surprisingly enough, they're actually pretty common in the human population and indeed in biology in general, which is kind of surprising when you think about it. So studying DNA using a microscope seems kind of bonkers in today's day and age, but cytogenetics, which is this field studying DNA sequence with a microscope, is still done sometimes. Because even though we have very fancy and sophisticated modern techniques for sequencing DNA, actually catching large changes is sometimes difficult using those methodologies. So what this type of geneticist will do is essentially extract DNA from cells, stain that DNA, and look at it on a microscope slide. Normally, that person will be looking at metaphase chromosomes. And the reason is, as you guys know from the work that we've done on cell division, and indeed, if you look at the sort of bottom right-hand corner of this slide, upon metaphase of meiosis or mitosis, the chromosomes are very condensed, meaning they're not long strings anymore. They've kind of brought themselves into a condensed state. And they've been replicated as well, and they generally appear like X's. So you see some metaphase chromosomes from humans, Drosophila, and from corn on this slide. And that's often what people think about when they think about pictures of DNA. But now you know what you're really looking at is metaphase chromosomes of organisms. And again, it's just because they're easy to visualize and look at because they've been condensed and they're easily separated. A karyotype is essentially what you're seeing in this slide, which is just stained chromosomes from an individual. A karyotype specifically is all the chromosomes from a single cell arranged in a standard way. So it lets you look at your entire genome for that cell under the microscope. Oftentimes when geneticists do this, they stain the DNA using a geasma stain, it's called, which leads to something called G-bands on the chromosomes. So on this little inset, you see standard staining here. This is just staining for DNA. Underneath that, you see G-banded chromosomes, which have just been stained with a different kind of stain. And it just creates a very specific banding pattern, which you can use to actually look at chromosomes. So cytogeneticists look at banding patterns produced by chiasma staining. They look at size of chromosomes, and they also look at another common feature of chromosomes, which is where the centromere is presented. You can see that most easily really on this kind of middle set of chromosomes in the standard staining. If you look at this thin pinching in of the DNA, that is where the centromere is in that particular case. And you can actually designate where the centromere is using different letters, different words. So just to remind you what a centromere is, if you look at this subset, this little inset over here, you see the centromere is basically, it's a protein structure that sits in between the two sister chromatids, attaching them together at metaphase. So remember, one chromosome is these two sister chromatids, which are just replicated single chromatids. And the centromere holds them together, and it's also where the microtubules are going to attach to kind of pull the two versions of this chromosome apart. So if the centromere is smack bang in the middle of the chromosome, it is called a metacentric chromosome, as shown here. If the centromere is just off center, it's called a sub-metacentric chromosome. If the centromere is relatively close to the end of the chromosome, it's called an acrocentric or acrocentric chromosome. And in some cases, the centromere is right at the end of the chromosome, and that is a telecentric or telocentric chromosome. Please notice as well that each different chromosomal arm, which just means either side of the centromere, is either designated the P arm or the Q arm. P stands for petite or small in French. That is always designating the smaller of the chromosomal arms. So you see in this acrocentric chromosome, P is the little arm, same with the submetacentric over here. And Q is just, and this is slightly embarrassing as a scientist, but Q is the name of the other arm, and it's called that because it is the next letter in the alphabet after P. You'd think you'd go for G for, grand, but no. So Q and P are the two arms of the chromosome. Now, this is a karyotype of a diploid human cell in the top left-hand corner here, so 2 copies of each chromosome. You can see in this nice karyotype where the centromeres are. This one looks like it's a metacentric one. In 13, 14, 15, they look acrocentric to me. And indeed, all of the banding patterns that you see here have been named and numbered for cytogeneticists, and that's what's shown down here in this schematic of all the chromosomes found in human cells. You can diagnose certain genetic problems using karyotypes and look at how chromosomes differ between different species, among other things, and that's why they perform these kinds of experiments. So that's sort of how you can examine human karyotypes and look at chromosome structure writ large. And what we're going to talk about for pretty much the rest of today, or this lecture for sure, and also the next one, are the ways in which the structure of chromosomes can be altered, which can cause problems in the organism. There's 2 big ways that structure of chromosomes can change. The first is going to change the total amount of genetic material in a chromosome. The first of those types is deficiencies or deletions, when you lose part of a chromosome. The second type is called duplications, where you basically duplicate part of a chromosomal segment. The second main way that chromosomes can be changed is that the amount of DNA, the amount of genetic material remains the same, but you rearrange it in some way. One of those types is called inversions, where you basically flip around part of a chromosome. Basically, you break it, flip it around, becomes reconnected. And the second is a translocation, where part of one chromosome breaks off, becomes attached to a different chromosome. And we're just going to go through these one by one to show you how they occur and the kind of phenotypes that can arise when they do occur. So relatively straightforward are deficiencies and deletions. So here on the left, you see a chromosome with, of course, the two arms, the centromere in the middle. And all we've done is number different parts of the chromosome so you can see what order they come in, and you can diagnose what happens when there's some issue with that chromosome. So here we have a simple deletion where we've had a single break occur in the left-hand side of this chromosome where we've lost segments 3 and 4. So now these become lost in the cell, they become degraded, and you end up with the end of the chromosome being missing. This is called a terminal deletion because you're deleting the terminal end of the chromosome. That's one possibility for a deletion where you just lop off the end of a chromosome. The second kind is called an interstitial deletion. And here we have the same chromosome, but what we've shown this time is we haven't lost the end, we've chopped out the middle part. So we've lost numbers 3 and 2 here. The number 4, the very end part of the chromosome, actually got joined back on and got repaired back onto the original chromosome. This is called an interstitial deletion, where you lose some central part of the chromosome, but you keep the end because there's a repair step. Now, deficiencies and deletions can have obviously important phenotypic consequences. And like most of the things we're going to talk about, the size of the change, just in terms of how much of the chromosome is gone or how much more chromosome you have, tend to have a large effect on any phenotypes. The bigger the size of the problem, the bigger the size of the phenotypic issues. So the size of the deletion matters because the number of genes that you lose matters. And what matters as well is, of course, if the genes that you've lost are actually important for the organism or not. Now, as you would expect, normally when there's a deletion and there's a phenotypic effect, it's usually detrimental to the health of the organism. One example of this is a disease called Cri-du-Chat syndrome in humans, and this is actually a deletion of the short arm or the P arm of chromosome 5. So on the right here, we see 2 versions of chromosome 5. You can see the centromere is right here, where the kind of pinching in is, DNA. So we're looking at the short arm, and we've lost, close to half of the short arm. If this occurs to an individual, then they have this syndrome, Cri-du-Chat. It's called that because Cri-du-Chat means cry of the cat in French. And as infants, people that have this syndrome, their cry when they cry sounds like they're a cat crying. And it's associated with other phenotypic abnormalities, such as facial abnormalities and mental retardation. And you can kind of see some of those facial abnormalities on this little girl on this slide. So deletions, you lose part of your DNA. Duplications, you actually end up duplicating part of a chromosome and somehow keeping it. It can happen by different mechanisms, but often it's caused by abnormal events during recombination. So you know that recombination occurs as part of the mixing of DNA in meiosis, in meiosis 1. And recombination is driven by the same sequence of 2 homologous chromosomes, like 2 versions of chromosome 1 that you got from mom and dad. They line up by virtue of the fact they have very similar sequences, and then there's a crossover between the 2. Sometimes they don't line up correctly, as is shown in this example on the right. So there's sometimes repetitive sequences present in a chromosome, and in red here is essentially very similar sequences, but they're repetitive. So when these two chromosomes line up, normally the first red repetitive sequence would line up with the first red repetitive sequence in the homologous chromosome, and they'd line up perfectly normally. In this case, that's not happening, and you can see that they are misaligned because these repetitive sequences have lined up to the wrong repetitive sequences on the other chromosome. Now, when this crossover happens, you can see what kind of thing is going to occur, right? You're going to get this large part of this blue chromosome containing C and D, which is going to get swapped with the end of this chromosome, which just contains part D. Now, if this occurs during crossover, you're going to get the following four possibilities in gametes. You're going to get a normal purple chromosome at the top, a normal blue one at the bottom, and then these weird ones in the middle are because of the misaligned crossover. And what you can see now in this really long chromosome is that we've duplicated the C part of this chromosome. And if you look at the other chromosome, we've actually deleted part of it, right? Sorry, this one down here. We've deleted part C of this one. So this is how you can get deletions and duplications occurring. And duplications simply add a chunk of DNA to the chromosome. It is often true that duplications have less of a harmful effect than deletions of comparable size, and that kind of makes sense when you think about it, because you're getting extra DNA, not taking it away, although that can also be detrimental. Again, size matters here. The bigger the rearrangement of the chromosome, the more likely you are to have some kind of phenotypic effect. Smaller chromosomal duplications don't tend to have any phenotypic effect at all, but they're very important from an evolutionary standpoint, and here is why. If all of a sudden you have an extra copy of a gene, then mutations can occur during, over tens of thousands of years of evolution that change the function of one of those duplicated genes, but not the other one. And that can lead to a new function for the duplicated gene. And this is how you get things that are called gene families, where you get genes that are very similar in sequence, but have different functions in the cell. And that's because there was some duplication event a long time ago in evolutionary history, which caused, two genes to have exactly the same sequence and exactly the same function. And then over evolutionary time, one of them picked up mutations that gave it some extra function in biology. In case you're wondering, of course, it's possible for a duplicated gene to pick up mutations that stop it working. That's also possible. So duplications can lead to gene families, which give genes that are very similar, but have distinct functions from each other. These two genes that are derived from a common ancestor are called homologs. And when they're within a single species, they're called paralogs. So you can have homologs between species and paralogs within species. To give you a cool example of this in human evolution, you can use basically globin genes, so beta globin, hemoglobin genes to show you how things changed over time. So what you're looking at here is the modern versions of your hemoglobin genes at the top here. And then as we go down in this slide, we're going back through millions of years of evolution until we get back to the single gene that a bunch of genes in the globin gene family came from. So millions of years ago, there was one ancestral globin gene. There was some kind of gene duplication event, where part of the genome was duplicated. That gave us 2 copies of this gene. One of them went on to become myoglobin. That's the type of special globin gene that's in your muscles. And the primordial version of hemoglobin then split into yet more genes that gave more opportunities for evolutionary tinkering. So after the gene duplication occurred, one of the duplicates picked up mutations through evolution that made it better for functioning in muscle cells, and that became its job. And that's what you have today. You have the myoglobin gene that's active in all of your muscles that helps them store oxygen for physical activity. The second version of this gene became better at binding and transporting oxygen in your red blood cells. That's handy. We all use that every day to transport oxygen from our lungs to our tissues. That gene was duplicated again to give you alpha chains and beta chains. And now you actually have lots of versions of this gene that do slightly different things in your blood to allow it to carry oxygen. So for example, the purple stuff here, actually let's start with the red stuff. So the red stuff is a version of so hemoglobin, the protein that carries oxygen in your blood, is made of subunits. It contains 2 alpha chains and 2 beta chains. When you were very early on in your development in embryonic life, still in your mum's uterus, then these versions of the alpha and beta chains are expressed because they're really good at carrying oxygen early in embryonic life. Slightly later on in your life in utero, in the 2nd and 3rd trimesters of pregnancy, these purple versions of the beta chain are expressed because they work best for what you require at that point in your life. After you are born and you become a what a developmental person might call a fully formed organism, the original versions of this gene are shut down, and this green one, beta globin, is expressed because it's best for your survival as a functioning organism outside the womb. So the globin gene family is huge. It's resulted from several gene duplication events, and throughout evolution, they've all been kind of tinkered with. They've all evolved slightly different functions to allow, and they're expressed when they're required during your life at the appropriate time. Now, throughout the course of evolution, there's been many duplications and deletions in the human genome and in every genome. Because of that, there are parts of your genome that have what is called a copy number variation, or CNV, which means you might have part of your one chromosome where you're missing some genetic information, and then you might have part of another chromosome that has a duplication, so you have more information. They're actually small copy number variations are very common throughout biology. Generally, from 0.1 to 10% of a genome might show copy number variation. And your average human, it's about 0.5% of your genome, which is still a lot that shows copy number variation. And sometimes that can be linked to certain diseases like schizophrenia, autism. We're discovering that a lot of these diseases have something to do with carrying certain copy number variants. Here's an example of that. So here's some members of a species that have these 2 homologous chromosomes. If you inherit one chromosome with a copy number variant in it, this shows a duplication, for example. You carry, here you got this chromosome from your dad. There's 2 different versions of this part of the chromosome that we've called A. So, as I said, there's lots of different diseases that have been associated with copy number variation. Some of them are listed here: schizophrenia, autism, learning disabilities, certain cancers and infectious diseases. And that's all I have to say for this lecture. So please join me for the other lecture, where we're going to talk about some of those other chromosomal variations that you see. So see you on that one.